Hello everyone, welcome to our class. So today we are going to start a very interesting topic that is a type of cells. And we will take a closer look at a prokaryotic cells. So we are going to learn about prokaryotic cell. So let's begin. Now listen carefully. On the basis of cellular organization and the presence or absence of the nucleus, cells are divided into two main types. The first type is called as prokaryotic, which have a simple cellular organization, and the second type is eukaryotes, which have a higher and more complex cellular organization. So, in today's class, we will focus only on prokaryotic cell, that is simpler ones, and understand their structure in detail. Prokaryotic cells. Prokaryotic cells, also called as a primitive cells, means pro means primitive, karyotic means we can say nucleus. Okay, primitive nucleus cells. It's called as a primitive cell. So why primitive? Because they are considered the earliest or oldest form of the life on Earth. The most important feature that is a prokaryotic cell do not have well-defined nucleus. Their genetic material is not enclosed within a nuclear membrane. Instead, it just floats freely in cytoplasm, as we can see in this image. There is a no evolved or advanced type cell organelle present. Either no genetic material is no bounded by any nuclear membrane. So basically, we can say that nucleus is absent in this prokaryotic cell, or we also said that nucleus is not properly or definite in shape or there is no also histone protein in large amount present. That's why, as we can say, okay. Let's their genetic material is not enclosed within a nuclear membrane, as we can say. Also, remember, these cells do not have membrane-bound cell organelle like mitochondria or chloroplast. So everything happens in their in the cytoplasm in this cell. Let's now talk about the genetic material. In prokaryotes, its genetic material presents in the cytoplasm and is also known by several names. You can call it nucleoid, genophore, or sometimes prochromosome. This DNA is circular in shape, double-stranded, and haploid. Haploid meaning there is only one set of gene is present. And here something is interesting that some bacteria have small extra piece of DNA called plasmid. Certain bacteria also have extra chromosomal DNA other than main genetic material. These plasmid are not part of the main DNA but carry extra genes. For example, this gene that give antibiotic resistance. Okay. Okay, now let's talk about next structure of this prokaryotic cell. Let's move on the cell envelope. As we can see in this image, uh, here is a protective covering of this bacterial cell or prokaryotic cell. Mostly prokaryotic cell present in only bacterial cell means we can also say that bacterial cell have or bacterial cell is made up from or bacterial cell have prokaryotic cells, a simpler one. Okay. Let's talk about cell envelope. Uh, this is basically the protective covering around the prokaryotic cell. It is a quite complex and plays a major role in protection and structure. The cell envelope is made up of three layers. It consists of three layers. That is, outer layer, outermost layer is called glycocalyx. Middle layer is called cell wall, and innermost layer is the plasma membrane. So let's go one by one. First is glycocalyx. As we can see, this is a glycocalyx. Okay. So Glycocalyx is the outermost covering. It can appear in two different forms. That is a one is a slime layer, which is a thin and loose sheath type. Okay, and other one is a capsule, which is a thick or a tough covering of this uh, uh, prokaryotic cell. When the capsule is present, it helps the cell resist drying, and we can also say that digestion and protect it from harmful chemicals and immune attack. So it's like the cell's safety jackets, we can say. Okay. Next comes the cell wall. The middle layer is cell wall. Okay. Cell wall which form the middle layer. It is a thick and gives mechanical it is a thick and gives mechanical strength to the cell. It prevents the cell from bursting due to the osmotic pressure. The cell wall is mainly made up of substance called peptidoglycan in gram-positive bacteria and murine in gram-negative bacteria. So remember, the composition of the cell wall can vary among the bacteria. Now the innermost layer. Okay, this is the innermost layer. Okay. The innermost layer is the plasma membrane. It is made up of phospholipid bilayer, just like in eukaryotic cell. The plasma membrane plays key roles. That is, it provides the protection to the cell, and it helps in transport of material in and out of the cell. So it also known as a intracellular transport. So basically, it acts as a gatekeeper for the cell, as we can also say that. Okay, this is a cell envelope. Okay. Uh, Bacteria are better observed when stained, and that is a gram staining method was developed by Hans Christian Gram in a cell envelope. This is for the cell wall. Okay. Next, move to the next structure, that is a flagella and cilia. So some bacterial cell may be motile or non-motile. Okay. This is a flagella. And okay, this is a flagella. Okay. As we can say, some bacterial cell may be motile or non-motile. Motile cells possess flagella or cilia, which helps in locomotion. The flagella are driven by the rotatory movement produced by the basal body and are composed of three parts. That is a basal body, hook, and filament. So in this image, we can see that is this is a structure of flagella, and flagella consists of three parts. That is this part we can say it's a basal body. Then this is a hook, and this is a filament like structure. We can say it flagella. Okay, this flagellum show rotatory movement, and it helps to this cell to move or locomote. Okay. Uh, right. Next structure is pili and fimbriae. Okay. These are the surface projection on the bacterial cell. Pili help in intracellular communication or DNA transfer during the conjugation. So this is a pili or fimbriae. Pili and fimbriae. Fimbriae and pili are basically similar structure or okay. Fimbriae helps the bacteria cling or attach to surface and host tissue. Okay, let's move to the next structure. That is a mesosomes. So in this image, we can see this plasma membrane infolded inward. So this tube-like structure called mesosomes. Okay. The plasma membrane in some prokaryotic cells show infolding called mesosomes. This infolding, okay, this is called as a mesosomes. This structure help in DNA replication, cell wall formation, and respiration. This mesosomes is we can say that homologous to mitochondria in eukaryotic cell. Uh, Next structure is a chromatophore. In photosynthetic bacteria, we find the structure called chromatophores. These are a membrane extension containing photosynthetic pigment that help in a photosynthesis. Okay. Next one is ribosome. Prokaryotic cells have 70s ribosome, which is made up of 50s large subunit and 30s small subunit. Okay, these are responsible for protein synthesis inside the cell, and this all area in in cell, this is a area is called as cytoplasm. So cytoplasm is a semi-fluid matrix where all the cellular activities takes place. It contains ribosomes, nucleide, plasmid, and various enzymes. And in previous image, there is a some information about gram staining. Now, bacteria can be better observed when stained. The most common method used in gram staining, developed by Hans Christian Gram. This technique helps in classifying bacteria into gram positive and gram negative based on their cell wall composition. Okay, this is about gram stain. So, in this slide, we can study about, or we can see the difference between prokaryotic and eukaryotic cell. So, let's summarize the key difference between prokaryotic and eukaryotic cell. In this column, we see the features of cells, and here is a one column about prokaryotic cell, and here is a eukaryotic cell. So, first character we can uh, talk about, that is a nucleus. So in prokaryotic cell, a nucleus is not well defined, and eukaryotic cell there is a nucleus is a well defined shape and proper genetic material is bounded by the nuclear membrane. So in prokaryotic cell, nuclear membrane and nucleolus absent, but in eukaryotic cell, nuclear membrane and nucleolus is present. So basically, eukaryotic cell is a highly evolved or had a highly advanced type of cell. We also say that eukaryotic cells develop or evolve from prokaryotic cell, but that's okay scientifically. Okay. Next structure is DNA structure. In prokaryotic cell, DNA is circular, double-stranded, and naked. And eukaryotic cell, there is a DNA is a nuclear, double-stranded, and enclosed within the nucleus. Another one is character is a membrane-bound organelle. In prokaryotic cell, mitochondria, chloroplast, Golgi bodies, or nucleus are the membrane-bound cell organelle, which is absent in prokaryotic cell and present in eukaryotic cell. Ribosomes, in prokaryotic cell, 70s type ribosome present, and eukaryotic cell, 80s ribosome, 80s types of ribosome present. Cell size is small in prokaryotic cell, usually 1 to 10 micrometer, and eukaryotic cell, it is a large and varies from 5 to 100 micrometer. Cell wall composition. In prokaryotic cell, cell wall is made up from peptidoglycan or murin. And eukaryotic cell, it is made up from cellulose in plant cell, chitin in fungi, and absent in animal cell. So, example of prokaryotic cell is a bacterial cell, and eukaryotic cell is example of eukaryotic cell is a protista, fungi, plants, and animals. So this is all about difference between prokaryotic and eukaryotic cell. So in this we can summarize what we can learn in this topic. Okay. So basically, prokaryotic cells are simpler and smaller, but they carry out all essential life functions. They form the basic and ancient design of life, from which more complex eukaryotic cells evolve later. So let's see. So definition of prokaryotic cells, that is, it is a primitive cell without a well-defined nucleus or membrane-bound organelle. Best examples are bacteria and cyanobacteria. Let's talk about the structure of prokaryotic cell. So, genetic material present in cytoplasm, as we can say, as we can known as a nucleoid. It is a circular double-stranded DNA. Some bacteria also have extra chromosomal DNA that is called a plasmid. Cell envelope has three layers, that is a glycocalyx, cell wall, and plasma membrane. Glycocalyx is outermost, a cell wall is a middle, and plasma membrane is an innermost. Other components like flagella and cilia helps in movement, villi and fimbriae help in attachment and intracellular communication. Mesosomes, it is an infoldings of plasma membrane and helps in the DNA replication and respiration. Chromatophores contain pigments for photosynthesis in some bacteria. 70S ribosomes are present, which is help in a protein synthesis. Cytoplasm is a semi-fluid matrix where all cell activities occur. Okay. Cell wall staining. It is better observed when stain using Gram staining method, which is developed by Hans Christian Gram. And we saw the difference between prokaryotic and eukaryotic cell. So prokaryotic cells, no true nucleus, 70s ribosome, peptidoglycan wall, and very small size. In very small size. And another is eukaryotic cell, which is has true nucleus, 80s ribosome, cell wall made up from cellulose or chitin if present, and is basically larger in size compared to prokaryotic cell. Okay, thank you.